Okay. So in this chapter, again, we're finishing up 15, which is the adaptive. So I was having a couple questions asked before class, as well as over emails, in terms of how to process all this material. Chapter 14, innate system, right? So think about it from a standpoint of where's the beginning. The beginning is you're healthy, you're fine. There's going to be some sort of infection agent that gets inside. Bacteria, viral, fungus, it doesn't matter. What are the first levels of protection is your innate system, right? So your innate system is your skin and your mucus mainly. Then you got the PRRs underneath there, the pathogen receptor, pathogen recognition receptors, right? That's looking for all the things that we have evolved to know that are bad. Peptidoglycan, LPS, double-stranded RNA, flagella, things that our body doesn't normally have. And so to have those be recognized by the PAMPs, or by the PRRs, they are then send out signals, distress signals. What are those distress signals? Cytokines, chemokines, interleukins. Remember, those are the voice. Those are what the cell uses to talk to each other. Then, if it's not able to clear the infection, it can go kick on complement. Complement's a bridge between the innate and the adaptive. Remember, the adaptive takes about 10 to 14 days to come completely online. That's because in the adaptive system, now those macrophages, those dendritic cells that went to the site of the infection, they phagocytize, they process them, put them up on their MHC class 2s. They go back to the lymphatic system. They get in the lymphatic system. They go try to meet their B and T cells. If it is a T cell dependent antigen, which is pretty much almost everything, it needs to have the T helper cell, which is the CD4 cells, needs them to go help, to go tell the B cell, yep, that's a good one to go after. Once the B cell gets that signal, it matures into a plasma cell. The plasma cell is what's actually producing the antibodies, right? So only plasma cells produce antibodies, that's it. But it requires all that extra help. T independent, which is right here, is if the antigen that the B cell comes in contact with is one of those PAMPs, one of those LPS, peptidoglycan, double-stranded RNA, whatever it might be. It doesn't need any help because it's already evolved to realize that that's something bad, and that it knows that it's something it needs to go after. So it doesn't need the T cell to come over and say, "Yep, good job," because it already knows it's a good job. It knows to go after it. So that's why we call it T-independent antigens. Those are ones that do not need the extra T cells' help to actually start the immune system to start producing the B cell to move on to a plasma cell to start producing antibodies. So LPS is another T-independent antigen, as we already pointed out. So it's just telling you LPS is one of them. Up here, molecules with numerous identical, evenly spaced epitopes are bound by a cluster of B cell receptors. Leads to activation. Not very immunogenic in young children. So the T-independent process isn't very good in children. So again, what are the children relying on? The maternal antibodies that last for the first 6 months of life, right? Before the immune system comes on. So these T-independent antigens aren't going to work very well in kids. And when I'm talking kids, I'm talking infants. Because again, their immune system isn't online yet. So even though their immune system recognizes through genetics and history, it recognizes that these are bad antigens, the immune system is not completely up and online, and it can actually do much about it. Of course not. All right, so switch here. So here the T cells play different roles in the B cell, right? They never produce antibodies. Just get through your head that it's only the plasma cells. Plasma cells are only the B cells that have become effectors. The effectors are the ones that are doing their jobs. It just happens to change name to a plasma cell, even though it's still a B cell. So remember, there's two major types of T cells that we're focusing on in this chapter. We have the CD8s and the CD4s. CD4s go after the MHC class 2s only, right? So it's the C helpers. The T helper cells are the CD4s. So the CD4 T helper cells is the full name. They're going after the MHC class 2s. The MHC class 2s are only found on antigen-presenting cells, which are your macrophages, dendritic cells, and also B cells can present. The CD8 cells are going after the MHC class 1s. The MHC class 1s are found on every single nucleated cell in your body. So these are going to be the ones that go after the cells that are infected with something internal, usually a virus. So the MHC class 1s can send a distress signal up. Normally, MHC class 1s are just sitting out there as an address label, so the cell recognizes self versus non-self. But sometimes the MHC class 1 can take antigen that it finds inside the cell and put it up there as a big red flag, saying, hey, not only hey who I am, but hey, here's what's inside me. Somebody help me. Unfortunately, that help is going to come in the form of CD8 cytotoxic cells that are going to come and ask that cell through cell signaling, tell that cell to go through apoptosis to essentially kill itself, to minimize the chance of that virus pushing on to continue spreading throughout the body. So here we're just talking about the T cells only, general characteristics of both CD8 and CD4s. We have the two polypeptide chains that essentially act as its antigen receptor. Don't worry about beta, gamma, and delta. That's all above the necessary means of what you need to know for this particular test. The book gets into the weeds a little bit on that, but recognize that there are structurally comparable to the B cell receptor, because what is it doing? It's recognizing the same antigen. Antigen is going to be what both the B cell and the T cell receptor has to recognize. And so in this case, it's going to be similar. That makes sense because they're recognizing the same antigen. The T cell receptor does not interact with free antigen. All right, so what does that mean? The antigen has to be displayed in the MHC class 2 or class 1. It just can't find antigen floating around in the body and say, oh, that's what I want to go after. It has to be in that MHC class 2. B cells can recognize free antigen. You'll notice that if you go back and look at the slides before this one, you'll see that it says B cells can recognize both free and processed inside an MHC class 2. So in this case, there's a little bit of a difference in the T cell. So that's what it's saying here, only antigen presented by inside the antigen presenting cells. And the antigen has to be displayed in that MHC, right? In this case, it has to be an MHC class 2. If it's looking for the CD4 T helper cells, it has to be an MHC class 1. If it's recognizing the CD8 cytotoxic cells are going to recognize it. So here's what that says already. Another way to think about it, as I've already pointed out, the MHC class 1s are for endogenous. So endo means inside. So those are antigens that are already inside the cell. Usually going to be viruses, but occasionally it can be toxins. Occasionally it can be some very, very small bacteria. As I pointed out, the bacteria that causes chlamydia is an intracellular bacteria, which is kind of odd, but we'll see all that as we get up into the different chapters. MHC class 2s are presented in exogenous, so outside the cell. So when we talk about endogenous and exogenous, endo means inside the cell, exo means outside the cell. MHC class 2s focus on antigens outside the cell. MHC class 1s focus on ones inside the cell. Yeah.
So, what does it mean by, what is an antigen on the outside of the cell doing? Is it just like...
Think about bacteria. Bacteria is too big to get inside the cell. And so it's going to be floating around in our blood vessels. It's going to be floating around in tissue. It's not inside an actual cell. It's in the outside cell areas.
Okay.
But it's still, it's a bacteria. It's a fungus. It could be a toxin. It could be a virus.
Is it necessarily bound to its outside membrane, or it's just just not?
Yeah. Imagine she's a cell and I'm the bacteria. I'm just sitting here next to her, right? I'd be exogenous. A virus would be one that got inside the cell. And now it's causing conflict inside the cell. It's a way for the cell to get. Because once the virus gets inside the cell, it's really difficult for the immune system to recognize because now it's hidden. It's inside the cells. And so it has to be able to have a way to be able to show that it has a problem. It has that problem, it puts it up on MHC class 1. So characteristics continued. CD8 T cells recognize antigen presented in MHC class 1. So hopefully by the time this is all done, you've listened to the videos, you've got this process in your head. Like I said, this is two of the questions that people tend to miss the most. I think they're gimme questions, but they tend to miss them. Is like, what does CD8 T cells recognize? MHC class 1, CD4s. So that's what we see right here, same thing. Cluster differentiation, just telling you what the CD stands for. There's hundreds of them. We use them as immunologists to differentiate all the different cell types. If you've ever looked, and I know you all have, look at cells underneath the microscope, they all look pretty much the same. right? It's really hard to differentiate a cuboidal cell from a squamous cell from a columnar cell sometimes. And that's just talking about the three different types of epithelial cells. Imagine all these white blood cells. Remember, all these macrophages, dendritics, T, B, these are all white blood cells, all of them. They all pretty much look the same underneath the microscope, except for some subtle differences. So how we're able to recognize the different cell types is based off of these different markers that we've discovered. We use fluorescent antibodies, like we've seen before with the fluorescent microscopes. We can also use those same antibodies to do something called flow cytometry, which is a way to count the different cell types. It actually goes through a machine. It looks for those fluorescent light ups that we've seen before with the microscope pictures. It looks at those different lights that go off as the cells go by with the different tags we put on, and it's able to count the number of cells of different types. And so that's why it's called cluster and differentiation. It's a term we gave. It's a way that we're able to differentiate the different cell types. And so, again, there's hundreds of them. Every time we discover a new cell type or we discover a new surface marker, it's probably going to be given a CD name as well as a functional name. So whatever that particular marker may do, it may be called CD 220, and it may have a name like, dendritic cell marker 3 or something. It's just going to be, sometimes we give them both names just as a way for us to keep track of them all. So there's actually posters you can get from some of these immunology companies that sell all these different antibodies that will give you a poster of all the CDs on it and what they actually bind to. So in this case, just recognize that that's what they are. It's just the way that we are able to differentiate them. We just happen to have discovered that CD8s and CD4s are important T cell markers to help differentiate the two major types. As we already talked about last time, CD4 is the receptor for HIV. So this is why and how it's able to cause disease, and then ultimately how it actually causes somebody to succumb to the disease. You get infected with HIV, you die of AIDS, but ultimately you're dying of those secondary infections, right? Because your immune system has lost all its CD4s. The virus has killed off all the CD4s. There's no helper cell there now. It can't activate the immune system, and therefore any sort of disease that you get, your body has no way to fight it off, and it eventually just takes you out. The other receptor that we've discovered, just for you to know, is called CXCR5. So it even gets even more difficult than the CD markers. They get these other names. So the two major markers are CD4 and CXCR5 that HIV actually attacks. Just goes to show, and what I want you to point out here is, help you understand how different diseases work and how these things are important, but also to help you understand that viruses have to find that receptor. Viruses are only going to be specific to certain receptors. They're not infecting all your cells. They're only infecting the ones that they're actually able to recognize a receptor. When it binds to that receptor, the receptor gets a false information signal to phagocytize that virus and allows the virus easy entry into the cell. So things like hepatitis only infects liver cells, right? So we got hepatitis B, C, A, D, and E, actually. They only affect the liver cells because that's the only receptors it's able to recognize. So just keep that in mind, too, that viruses and bacteria are very specific in what cell types they're actually going after. Right. Diversity. So this is talking about how do we end up with all these different capabilities to recognize antigens. How is it that we can have diseases that are brand new? I mean, just in our lifetime, whether you're my age or you're a little younger, we've had multiple emerging diseases that have shown up on the scene. West Nile, SARS, just to name two of them. Ebola came back out again. Lassa fever came out. These are Chikungunya disease is one that they're fighting right now in Africa that's starting to spread. These are all new viruses that humans have never come in contact with. But how is it that a lot of us already have a natural immunity to them? Well, it's going to be because of the diversity and the capability of us to develop and recognize antigens no matter what they are. So first, we're talking about the diversity in the MHCs. So ensures proper presentation of the from the evolving pathogen populations. What that's just referring to is all these different types of new diseases that are showing up on the scene. So what you have is you have your set that you remember you get half your genes from your mom, half your genes from your dad, whether you like them both or not. You got their genes. And so we have HLA. I'm not going to ask you any questions on HLA, but if you want to know what it stands for human leukogen. What is it? Leukotriene, yeah, leukocyte, leukotriene, something a little bit different. Antigen. So it's just HLA is the old way of referring to MHC. We use MHC now. So all of you going into the field from this point forward are only going to use MHCs. But historically, they used to be called HLAs. So the gene names still tend to keep history associated. So sometimes it becomes difficult as you've already recognizing, there's a lot of stuff to figure out just in these 2 chapters. It becomes even more problematic when they're using historical names versus the common names. But again, because these are referring to the genes, they're named the historical name. So you can cross out HLA and put MHC there. It's the same difference. But notice here, this is going to be from mom. This is going to be from dad. There's 3 major genes that you can come together and make your MHC. Remember what an allele stands for. An allele is different variations of that gene. So think about like eye color. How many different eye colors are blue, green, hazel, brown? Lots of different ones out there. But you only got one gene for eye color. But the different alleles are going to be blue, green, brown, hazel, right? The different capabilities that gene has. So here, for our HLA-B gene, 1,400 different possibilities that you could inherit from your mom. 620 different possibilities for gene C. Gene A has 890 possibilities. Hopefully, even if you're not strong in math, you're recognizing having that many different possibilities just inherited from mom. You're only going to inherit one of those. You only get one of the 1,400, one of the 620, one of the 890. But when you do that math, that number becomes extraordinary for somebody else to inherit that same pattern. Then you have to think that you also get half of them from dad. And so you got those same possibilities. One of 1,400 from dad, one of 620 from dad, one of 890 from dad. Those 6 come together to give you your MHC. Remember, everybody's MHC is a little bit different, right? We're actually starting to do things like MHC typing now. I think I pointed out before. Maybe it'll make more sense now. We always do blood typing, right? We recognize that it's important to get the blood typing right because you can kill somebody putting the wrong blood in them. So we've already been doing blood typing for quite a bit. But now we're recognizing how important MHC typing is. Yes, the statistics is going to be incredibly difficult to find somebody that has the same combo as you, but it's possible because the number is still small enough that it's within a few million, 1 out of a few million people are going to have the same MHC as you. And like I pointed out before, I think you might have the need for a new liver or kidney one day. And to have MHCs be cared properly is going to be incredibly important. So that way those CD8s don't go around trying to kill your new liver. Right? If the MHCs are similar to yours or very similar or even identical, you're not going to have as bad of a transplant issue or rejection issue. But this shows us how it becomes such a very different number, how difficult that is to find a match. But now with the current technology of genetics and whatnot, it is something that we do. And so we're in those brand new phases within the last few years of not only typing your blood type, but also typing your MHC. So that way, if there is a time down the road that we can hopefully match you up as quick as possible, put all that stuff in a database, as a donor. And if you're an organ donor, that stuff will eventually go in a database. I don't think we're there just yet, but the idea is that we go into a database. You end up, succumbing, and your organs are ready to be transplanted. Goes in the database. There's a perfect match in North Carolina. Something we would never been able to do before, right? But we're going to hopefully be able to get this to this point. My clicker keeps giving up.
You said it was the CD8s that would eat the organs.
Mhm. Yeah, because remember they're going after the MHC class 1s.
Yes.
And so if the MHC class 1s are different, it's going to recognize it as a foreign entity. And it's going to want to try to take it out. All right, so activation, hopefully we've already covered a lot of this. This is going to be regurgitating it a little bit. Activation of T cells. So we have to have those those antigen presenting cells. Let's just use an example. Dendritic. It could be macrophage also, right? These are just going to be the antigen presenting cells that are critical to activating the T cells. They're going to begin migrating so that once they have their antigen, those antigen presenting cells will leave the site of infection. So let's just back it up one more time. You have say you just got a puncture wound. Say you stepped on a nail. You got a puncture wound in the bottom of your foot. It may have bacteria growing on it. Now that bacteria is in the sterile tissue of your tissue, right? It's going to cause inflammation. Inflammation causes the lymphocytes to come to the site of infection. It's going to start battle with the neutrophils dropping its granulocytes all over it, trying to kill it. Macrophages are going to come in, phagocytize, destroy it, put it up on its MHC class 2s. The macrophage has to leave now, right? It has to go to the lymphatic system. It has to go to the lymph nodes and to the organs that are part of the lymphatic system because that's where all the beta T cells are hanging out. Remember, we want to increase the odds. We want to increase the chances of them finding each other. The only way to do that is have them all housed in the same spots. And so you're going to have them housed within the lymph nodes and spleen mainly. And so it's going to begin migrating the secondary lymphoid tissues. It's going to have it all loaded up as MHC class 2, and it's looking for the T cells in there. So that way it can, hopefully find its right match. So once it recognizes, the T cell needs to do two things. It needs to recognize the MHC, or it needs to recognize the antigen inside the MHC, and it also needs to give a co-stimulation to basically tell the cell that that's the right thing to go after. So it's got to recognize it and then tell it that it's the right thing. If the pathogen is detected using toll-like receptors, you got to go back to chapter 14 and think about toll-like. Toll-like are only found on the lymphocytes. So if you go back and look at the toll-like receptors in chapter 14, you'll see that they are found on the lymphocytes. If the pathogen is detected, it's going to travel to the secondary lymphoid, just like you produce a co-stimulatory molecule, danger signal, and ultimately start the process. And this can be in MHC class 1 or class 2. And that's what it's saying here. The only difference between what we've already been talking about and toll-like receptors, what we've already been talking about can only be in MHC class 2s. If it's one of these camps, again, that's what toll-like receptors are going after, are those the LPS peptidoglycan, those generic ones. If it recognizes them, it can be in both a 1 or a 2, because those, again, are the evolutionarily important ones that we just adapted that are immediate emergency signals. Macrophages and B cells can present in MHC class 2s and produce the co-stimulatory signals. All right, so here's an example of both of them. So starting up here, you have the dendritic cell in the tissue. I'm just going to read it to you since it's kind of small type. It says dendritic cells in the tissue collect particulates and soluble antigens and then travel to the secondary lymphoid. Boom, same thing. Dendritic cell is now phagocytized, antigen, put it up on its MHCs. Over here, it has them up on a 1 and a 2. So in this case, it must have been a important one of the major ones. Presents it to the CD4 T cells and also the CD8s, because remember the CD4s are after MHC class 2, the CD8s are after MHC class 1. This dendritic cell happens to have the antigens up on both. The T cells recognize the antigen by the dendritic cell, producing the co-stimulatory, activating those T cells to then go and do their job. Over here, they're going to the dendritic cells recognizing presenting self-peptides or other harmless molecules. They're not going to get that secondary stimulation. This is why it takes 2 recognitions, right? Going back to the slide before, T cells get activated by 2 methods. It has to recognize the antigen, and then it has to give that co-stimulation. In this case, it's doing both. This case over here, it's recognizing. That's a good thing. It needs to recognize that it's self. Right, so it's recognizing that itself, but it's not going to give the co-stimulation because it doesn't want to turn those T cells on. It doesn't want them to recognize, or it doesn't want them to go after the self antigens. And so in this case, it's doing step one, but it's recognizing it as something that it doesn't want to go after. So CD8s, once they recognize that MHC class 1, they're going to be able to distinguish between self and non-self by doing that co-stimulation signal. And then if it does recognize that the antigen is non-self, the T cell is then going to induce apoptosis through two major roles, proteases. So again, ends in ASE. It's an enzyme. Protein, it's an enzyme that chews up proteins. So it's going to start producing proteases that are going to chew up the proteins of that cell, as well release cytotoxins that are going to be in the form of what's called perforin. Perforin pokes holes. It's a toxin that pokes holes in the cell membrane. Poking a hole in the cell membrane is like popping a balloon. Doesn't matter where you poke the hole in, it's going to destroy the whole balloon. So if you poke a hole in the cell membrane, you basically cause a cell to pop. When that occurs, of course, the cell dies. So again, remind yourself, MHC class 1s are going to be things like viruses, things that are inside the cell. It can be a few different types of bacteria. Toxins can also get inside the cell. And so these are going to be things that the MHC class 1 is going to recognize. It's going to induce the CD8s to become active. CD8s are going to come in any cell that has that antigen inside its MHC class 1. The CD8 is going to come, bind, cause that cell to die. All right, CD4s, we've already gone over most of this. CD4s are recognizing MHC class 2. The T helper cells help activate other cells by producing cytokines. So again, what is it doing? It's just helping. It's its name. It's not really doing a whole lot besides telling the B cell, yep, that's what you want to go after. The B cell gets that cytokine signal to turn into a plasma cell. The plasma cell starts producing antibodies. It can sometimes be used as a conjugate vaccine. So when we use the word conjugate, that means that we are combining something together. So conjugation is just binding. So here what we're doing is just showing you the example of how we can actually convert something that's T independent into the opposite. So we can make conjugate vaccine against T-independent antigens. So in this case, Haemophilus influenzae. It's a bacteria. We used to think we'll get into it when we get into the respiratory disease chapter. Haemophilus used to be thought to be the cause of influenza, which is where its name comes from. But reality, it's bacteria that causes pneumonia. So kind of an old name, but it means something different. It has the T-independent. It has a capsule. Capsule is part of those T-independent antigens. Reverse the T-dependent antigen by covalently attaching the protein. B cell responds to the capsule. T cell protein activates the B cell. So what we're doing here is we're basically tricking the immune system through this vaccine. We're using that capsule that's going to induce a response. We bind what we're actually telling the immune system to go after to that, because this is going to give a strong signal. We bind what we're actually going after to that, and now we developed a vaccine against something completely different. But we're basically tricking the immune system because the immune system's so good at going after that capsule that we're just taking advantage of what nature's already given us. It's given us that capsule, and we basically attach what we really want the immune system to go over. Like, let's say, this isn't the case, but we could use something like the new papillomavirus. We can basically attach what we actually want to go after, like pieces of that virus, to the capsule, inject that. The capsule is going to be taken up, recognized, but now we actually have the immune system going after the other virus that we really wanted to go after. So it's just an example of how we do these things. Not that you need to know exactly how it works, right? We're not there yet, but just giving you an idea that a lot of your vaccines take advantage of these things that we know about the immune system to then develop new therapies and whatnot from. All right, role of T helper cell and macrophage activation. Just as another example, all this stuff was explained in the very beginning of the chapter, but now it's going through piece by piece, slide by slide, showing you how all these things are an example. So we already talked about in the beginning of chapter 15 how the cytokines can do a lot of different things. So once the T helper cell recognizes what it's supposed to be going after, it can turn on the B cell, but it also has these other roles to play. The other roles are going to be activating the macrophages, Sure, the macrophages do their job on their own. But if I activate them and say, I definitely want you to go and find this, the macrophage becomes even stronger, faster, capable of going and phagocytizing whatever that pathogen happens to be. So in this case, the T helper cells are going to have a role in macrophage activation through those same cytokines. The cytokines are telling the B cell to turn into a plasma cell, telling those macrophages to become activated, become stronger and faster. And in this case, they're going to increase their size, metabolism, number of lysosomes. So all of those things are good for the macrophage, right? Because the macrophage's job is to phagocytize. So if I increase its size, it can hold more on the inside.
Is it the T cell that's increasing size or the macrophage?
The macrophage. The T cell is giving the signal. It's saying, hey, you go and become bigger and faster and stronger. The T helper cell is just producing those cytokines, and the macrophage is receiving them, getting that signal to become bigger, so it can phagocytize more. Metabolism is able to process faster. Number of lysosomes, that's important for the chewing up process, right? Those lysosomes are going to be what's actually doing all the chewing up. Producing nitric oxide and other toxic compounds also helps speed up the capability of it to destroy stuff. And so sometimes when we actually, unfortunately, it's hidden behind these pictures, you might have, it might be better on your slides. Under behind there, it's going to say causes them to turn, I think, into giant cells. And so macrophages that become activated, we call them either A, activated macrophages, or B, giant cells. And the reason why we call them that is if you take a blood smear of somebody that has activated macrophages, my goodness, they're like 4 or 5 times bigger than all the other cells in the blood smear. It's pretty easy to tell that they're giant cells. That's where they got their name from. All right. Yeah.
I'm trying to make sure the T cells are they recognize and stuff. The MHC classes, are those the receptors?
Those are receptors normally. And so inside that receptor, they're going to be holding the antigen.
And then the CD4s and the CD8s, I just see that they're next to.
So the CD4s are what's recognizing MHC class 2s. So it's a recognition receptor that's on the surface also.
So the T cell is recognizing the invader?
So the T cell is recognizing the antigen up on the MHCs. What's recognizing the invader is going to be the macrophages, the dendritic cells, or the B cells that have already phagocytized it. They phagocytize it, they put them up on MHC class 2s. If it's a cell that's been infected by a virus or something like that, so it could be any of your cell types, they'll put them up on an MHC class 1. CD8s come around and they see that the MHC has something inside it. The CD8s are going to recognize MHC class 1s. If they recognize it, they're going to tell that cell to kill itself because it's infected. Keeps that virus there from spreading elsewhere.
But the CD8s are the ones that cause apoptosis.
Mm-hmm. Yeah, they're going to kill itself, apoptosis, yeah. The CD4s are only going to recognize antigen that's inside a antigen-presenting cell. So dendritic cells, macrophages, and B cells are antigens. It's a lot. I get it.
It makes more sense now. I was just trying to.
Yeah.
Individually, I knew what they were. I was trying to.
Yeah, so that's the biggest part about this chapter, really, is putting it all together. Once you get it together, it starts clicking in your head, and you're like, oh, why was it so hard in the beginning? But yes, I get it. Give you all this information in 2 chapters. It's a lot. All right. So the role, did you have a question? The role of the T helper cells and macrophage activation, like I said, it's just one of the other things that the CD4s do once they have recognized the CD or the MHC class 2, they become activated. CD4 is doing multiple things. One, it's telling the B cells to become activated. It's also telling the macrophages to become activated. Then we got our last of the 3 major T cells, right? We have our CD4s, we got our CD8s, now we have NK T cells. It's usually just called NK cells. NK stands for natural killer. I just think they're probably the coolest name. So natural killer cells induce apoptosis in self cells. All right, so these are going to be the same thing as the CD8s, but there's a difference. It recognizes host cells lacking MHC class 1. All right, so why is that important? Well, I've already told you that MHC class 1s are essentially the address label that tells the difference between self and foreign, right? So let's say all your nucleated cells are supposed to have MHCs on, MHC class 1s on. Some viruses, like I said, it's always a strategy game between the pathogen and the immune system. Some viruses actually get inside your cells. The first thing they do is pull that MHC class 1 off the surface. It's a trick. It buys them time. They buy them a day or two, but they can sit there and multiply and can help spread their numbers across new cell types. But as a counterbalance to some viruses being able to do that, we have NK cells. The NK cells go around the body. If they find any cells that do not have an MHC class 1 on them, they have to immediately kill them. They don't care. They don't care if you're infected or not. If you're missing your MHC class 1, there's something wrong with you if you're a cell type, right? And so they don't bother trying to figure out what it is. They just go ahead and take it out. And so that's the biggest difference. The CD8 T cell will take out too. It does apoptosis also, but it's when the antigen is inside the MHC class 1 on the surface. NK cells look for cells that do not have an MHC class 1, even though they should. If it's missing, they kill the cell too through apoptosis. So both CD8s and NK cells are going to go after cells that are going after MHC class 1. So let's say we got a cell type, we can just call it a liver cell or something. It doesn't have to be a liver cell. I don't want you getting focused when you go home and go, oh, it's got to be a liver cell. Just a cell, right? So let's say it's got hepatitis. The cell has been infected. It's got virus inside it. The virus gets processed inside, gets put up on the MHC class 1. Right? CD8 cells are going to come around. Recognizes that antigen in the MHC class 1, sends a cytokine signal, cytokines, inducing apoptosis. Right, so the cytokines are then going to go because it recognizes MHC class 1, it recognizes that that cell is infected, it sends the signals to tell that cell to basically off the cell. Now, the other opportunity that happens, so we got another liver cell here. It's infected. The part of this virus's strategy, again, we're just talking generics. We'll get into specifics as we start moving through the other chapters. One of the strategies that some viruses use is they pull the MHC. Well, let me do it in the same color so we don't throw colors off here. They basically will pull the MHCs off the surface. Right, so now the MHCs are inside, or they're basically destroying. And so the cell has no capability of telling the immune system that it's infected. So it's a good strategy at first, right? The CD8s are going to come by, and they're going to be question marks, right? They're not going to see it. They're not going to be able to recognize it because that CD8 has to recognize MHC class 1.
So what's in the cell that makes the natural killers kill that one and not my other cells without the MHC receptors?
It's looking for the MHCs. If it doesn't find it, it kills it.
Right, but what if my other cells, like the other cells that don't have MHC?
All your cells.
They all have
Okay.
that.
Yeah, so that's the key piece. All your cells in your body have MHC class 1.
And if they don't, they're getting killed.
They're going to take it out.
So are the MHCs inside the cell now?
Well, they've either been brought inside or they've been destroyed completely. It's just viruses do
So they're
different
normally
things.
like outside.
So the CD8s are going to be like, I don't know what the heck to do. Normally they'd be outside. Yeah, they're receptors, so they're on the outside. Now the NK cells, look at that. Notice there's no MHCs on it. Now they're going to do that cytokine response. Right, so they're going to do the cytokine response and essentially cause the cell to go through apoptosis.
So do all my cells also have the MHC class 2 as well?
Only antigen presenting cells. So only your B cells.
But the
macrophages,
natural
and
killer cells can Okay.
They're not going after it because those are going to be the MHC class 2s. They're only going after the MHC class 1s. So remember the difference is every single cell in your body has MHC class 1s. Those are the receptors recognizing cell. Tells your body that's your cells versus something else. So they all have to have it. MHC class 2s are only found in antigen presenting cells. So in this case, having your MHC class 1 missing is just like being recognized as being foreign. The NK cell has the adaptation to recognize cells that are missing their MHC class 1s, knock them out. CD8 has to have the antigen inside the MHC class 1 to recognize it and knock it out. So it's really the difference between the two. Does that make sense?
Yeah.
Better?
Yes.
All right. If you got more questions, you need me to redo it a different way, I can try a different way, but that's the best way I can think of it is the CD8s have to have the MHC class 1 recognition. NK cells are looking for the MHC class 1 missing.
And that's
Here's something. All right. So remember that's also called antibody-dependent cellular cytotoxicity. It doesn't need antibodies. And why are we calling it antibody-dependent? Because remember, antibodies can bind to the outside. Remember that's called opsonization. And that can also cause cytotoxicity because if it recognizes those antibodies found all over the cell, it knows it's something bad and knows to take it out. In this case, it's antibody dependent because there's 0 antibodies involved here, right? There's nothing happening besides the recognition that the MHC class 1's missing. And when it's missing, then we'll recognize that.
is ADCC the whole process of it? Okay.
That's the name of the process that the NK cells go through. All right. Lymphocyte development. In this case, we're just talking about what we've already said. The B cells mature in the bone marrow. T cells mature in the thymus. Remember, they go through a process to make that happen where all of it starts in the bone marrow, right? That's chapter 14. That's the hematopoietic stem cells. They're producing all your new blood, both red, white, and platelet. Immune system, we're mainly just focusing on the lymphocytes, the white blood cells. So in this case, the B cells are made by the stem cells. They stay in the bone marrow, become mature. Remember, maturity is just when they receive their receptors, their B cell receptors that helps them recognize their antigen. They haven't recognized it yet. There's maturity in there. They basically developed, been having their B cell receptor put on their surface, and now they're free to go out and start circulating through the lymph nodes, through the spleen, looking for those antigen-presenting cells to bring the antigen to them. T cells have to leave the bone marrow after they have been processed or have been produced. Then they go up to the thymus where they're educated up there. So where does all the different types of B and T cells come from? They come from this gene rearrangement, similar to MHCs, right? We talked about MHCs there on the slide before, where we're saying that you have the 3 different genes that you inherit from mom, 3 different genes you inherit from dad, come together to give you your unique makeup. Here, this is how the B cells and T cells are produced. This is what the hematopoietic stem cells are doing. They're not making every single B and T cell the same. They're basically rearranging the genes, just like we've seen with the MHCs. The difference is your MHCs never change. Once you become a fertilized egg, your MHCs are set for life. You got your half from mom, your half from dad. Fertilized egg becomes you. Your MHCs never change. Your B and T cells have to constantly be changing in order to be able to combat all the new different diseases and whatnot you may face in your lifetime. And so remember that each B cell responds to only one epitope. One epitope is about 10 amino acids. Those amino acids, 10 of them, right? We did the math the other day. You have 10 amino acids, but you have 20 different possibilities for each of those spots. It becomes something to the tune of essentially 10 to the 20th power, which is 10 with 20 zeros on the end, or the different possibilities that can happen. In order to do that, we have to actually produce that many different T and B cells to be able to go after those combination of epitopes. So we have 3 major genes that are involved. And all I want you to know, again, don't get lost in the weeds of the genetics of how it works. I know your homework does a couple little things that may get you thinking that you have to know more about this than what I want you to know. Know this, what's highlighted, right? It's a variable, diversity, and joining are the name of the 3 genes. They are joined and deleted different sections of the DNA. By doing that, it helps change the number, what the epitope is that B or T cell is recognizing. It's called imprecise joining. Its nucleotides are added or deleted, causing all that diversity. So it's naturally causing mutations on purpose. That's what we're doing here. Normally, mutations are a bad thing. Normally, a mutation causes some major disease. So, for instance, of your 3.4 billion genes or 3.4 billion nucleotides that makes up your genome, one mutation, just one nucleotide, can cause things like cystic fibrosis and sickle cell anemia. But in this case, we're actually inducing mutations, but it's at the stem cell level. They're doing it on purpose. And so this next slide is going to show you that actually happening. And so what we have here is the different genes. And again, I don't want you focusing on this because I know it's going to make your head break. What I want you to focus on is the fact that there is all these different types of genes out there, the different pieces and parts. All we're doing is tearing it apart. Think about like Legos. Think about you got however many different Legos that makes up. And I can tear them apart. I can put them together into different shapes. I can leave some pieces out. I can use all the pieces. It just depends. But anything I do with those, I'm creating different shapes, but I'm still starting with the same material, right? In this case, I'm just creating different B and T cell receptors by adding and taking apart those different pieces and parts. And in this case, those different pieces and parts aren't Legos. They're genetic material. But we can take this part and add it to this part, subtract out that part. Now we got one T cell receptor. Do it a different way, we got a different one. That's where your diversity comes from. So this goes back to the idea of what I was explaining. It was your last time before where I said sometimes some people, like you see these movies where there's some major disease outbreak. There are certain people that are naturally resistant. Why? Just pure dumb luck that they already had the receptors ready to go. And when they got infected, their immune system was able to knock it out. They developed an immunity to it. Others, they may not have that ready to go. And so they're going to succumb. It's just basically that strategy match between the pathogen and your immune system. And so if you already have it ready to go, and it just so happens to be your dumb luck that you had the receptors ready to go already, then you're going to have a much better chance of surviving that infection. Somebody else may not because their receptors weren't ready at that point. Is there any question?
In the psychology class I took here, the teacher was telling us about how there's people who engage in risky behaviors, quote unquote, not necessarily a disease that you're just talking about, and they have less susceptibility to get sick or get illness from it. And she said it was a study they did because some of them had survived, their ancestors survived the bubonic plague. Have you ever heard of that?
Well, two things I have to say about that. One, people that get into psychology are usually in it for self-help. And two, there are actual viruses, so herpes. Herpes actually, herpes gets into your neurons and actually hides there. And when it does that, it's actually related to the same virus that causes chickenpox. But what it does is when it goes into hiding, so people get herpes breakouts, right? You get however many different breakouts occasionally. Well, chickenpox turns into shingles, right? Younger people, they've had the vaccine now, so they may not ever have this happen. But it's the same family. And so herpes, why do people get breakouts? Sometimes once or twice a month, sometimes maybe a few months between breakouts. It's because that virus comes out of hiding and there's some sort of stress, some sort of stressor. In this case, for people that engage in risky behavior, there is an actual understanding that the virus, because it's stuck to the neurons, actually controls that. It controls them to actually go because what's it helpful for the virus? It's helping propagate and move the virus along. And so it's a different way of saying the same thing, what they're saying, but mine's based on genetics. Again, I've always joked about that. My undergraduate biology department was right next to the psychology department, and so we joked about it being self-help. But also, right? I'm good at that. She says that pisses a lot of people off, so I'm good at that. All right. So, anyways, I probably should have leave that on the recording. The whole talk, right? So, anyways, the lymphocytes. This whole process right here is just telling you how it is that you actually have all these different capabilities. How is it that we can actually go after 10 to the 20th different possibilities of antigens? It's because at the stem level, at the stem cell level, we are having this capability of producing all these different types of random receptors. Is it on purpose? No, it's completely random. There's absolutely no rhyme or reason to why this moment they're going to make these receptors, and next moment they're making these other receptors, but know that you're making thousands and thousands of new lymphocytes every day. Every one of those is going to have a different receptor on them, so the possibilities start dwindling. All of a sudden, it's 10 to 20 becomes down to 10 to the second, 10 to the third, because we end up having so many different T cells and B cells being born every day out of your stem cell population. So why is it that people tend to be sick in the very beginning of life? Because their immune system isn't ready to go yet. So a lot of diseases tend to take out children very young. And a lot of diseases take out people very old, because as you age, your immune system is not as good at doing its job. In between, we tend not to have as much of a problem with infectious diseases. Sure, can you get sick? Yep, we've all been sick. Do we get better? Yep, because our immune system is functioning fine. It's in a very early age, very late age that we tend to have problems. All right, so last slide here is negative selection. So I just told you, we make thousands of new T cells and new B cells every day. And unfortunately, some of them are going to be against self because, again, that VDJ recombination is completely random. So it may be that the randomness produced a B cell receptor or a T cell receptor that recognizes self. We don't need that in our lives because that ends up turning into an autoimmune disease, right? So we need to get rid of those before they actually go out into the population. So what is it that they're actually being trained? B cells being trained in the bone marrow, T cells being trained in the thymus. What are they being trained for? They're being trained to make sure that they don't recognize self. If they do, we need to off them before we let them out into the population. So B cells are exposed to self in the bone marrow. If they bind to self, because there'll be some self testers in there, and so there'll be some self antigens inside the bone marrow. They'll test the B cells. If they recognize them, they basically tell the B cell to off themselves. So that's called negative selection. Removes most of the B cells that recognize self antigen. So if they recognize self antigen, we need them to go. Positive and negative of the T cells. They have to go through both. Positive selection. The T cell has to be able to recognize the correct MHC. CD4s have to recognize MHC class 2s. CD8s have to recognize MHC class 1s. If they cannot, they are useless. They cannot do their job. And so that's called positive selection. We're positively selecting the ones that can find their correct MHC. Negative selection is going to be the T cells that are recognizing self-peptides. So the same thing as the Bs. So the T cells have to go through both. They have to go through being able to recognize their MHCs properly, and they have to be able to not recognize self-antigen. The process, as it points out here, the process is so stringent that over 95% of the T cells get booted. Only a few percent, only about 5% of the T cells actually make it through the whole maturation process. The rest of them are booted because the process is so tightly regulated because we do not want them to recognize self, right? So why is it that, an example of a autoimmune disease that tends to come out after pregnancy? What is it? Anybody know?
Postpartum?
No, autoimmune. That's lupus. Lupus tends to pop up after pregnancy. Why? Because during pregnancy, you have a foreign body growing inside you, right? So your immune system is kind of being calmed down a little bit, being told, don't go after it. It's getting kind of confusing signals. Some of those B and T cells sneak through that recognize self. After pregnancy is finished, they're out there now. They've made it through those checkpoints. There's no stopping them now. They're going to do their job because they weren't stopped in the beginning. They weren't stopped in that maturation process. So then after pregnancy, you become, everything goes back to normal. It's just yourself. There's no foreign body inside you anymore. The immune system cells, unfortunately, are out there attacking self now. And that becomes problematic. So it's not always the same. It's not always, lupus, but lupus tends to be one that we see in autoimmune diseases that develop after
So,
pregnancy.
because they didn't go through apoptosis or whatever when they were supposed to.
When they were supposed to, because again, the immune system's already confused because being told to ignore the baby, and so the immune system's already being thrown off a little bit. Sometimes these B and T cells sneak their ways through. They can recognize self. Just examples, again, trying to show you how this stuff all matters and how it comes together into the greater picture of what y'all want to do, right? Y'all want to go into allied health or nursing or doctor or whatnot. Help you try to understand why this stuff's important.
So is that why lupus is primarily seen more in females?
It's almost 100% female. There are females out there, but it's almost 100%
My
female.
cousin was male, he was 5 years old and died at 35 in complications.
He would be very
So
unique.
one of the very few.
Yeah, her question is why lupus is mainly in females. Absolutely. All right. Well, so as I said, see, I wasn't too bad off on deciding to postpone your test. So Thursday and Tuesday will be chapter 16. So go ahead and take a look at it. All of chapter 15, all the recordings, as long as today's went well, are going to be up online. So take the weekend to go looking through them.
So the test is gonna be
Next Thursday. This Thursday and next Tuesday, we'll just be doing chapter 16. If you didn't sign the sign-in sheet, make sure you come up and do it, or wherever it ran off to, right there. If you came in a little late, make sure you get your attendance.